Okay, so we start. Welcome everyone to this webinar, and I'm just going to keep it as short as possible because we need to leave room for the presenters. I will just introduce shortly what this is all about. First of all, this is connected with the HoliSoils, which is a project that is recently ending and sponsored by IUFRO, which is it's an organization that I'm not sure if everyone know of. In case you don't, it's a rather big umbrella organization of forest research organizations and institute. Their scope, it's mainly connecting all stakeholders involved in forest research. It's not only at the level of pure research. It offers some interesting opportunities for networking. And you see it's quite spread. HoliSoils, it's another part of the sponsors of the event. Keeping it short, actually the main news here is that we will soon release a greenhouse gas data set, which is quite a lot of measurements, several thousands, connecting greenhouse gases with microbial data in European forest sites. And just keep an eye on it. Over the next 6 months or 7 months, everything should come out. And there is definitely information to take out from it. And now to the task force, specifically to this task force, IUFRO Task Force. We are trying to gather people to work on the connection between microbial communities and biogeochemical cycling, specifically soil organic carbon is going to be the main point. So we work on all ecosystem services connected with soil organic carbon, trying to understand if and how they are influenced by microbial communities. And it's in science, it's maybe 20, 30 years that we speak heavily about it, and still quite a, I would say it's an edge of research, even if it's many years that people are working on it. This decomposition as an emergent ecosystem property was a big citation classes from 2011, but it was already there. The key research priorities that we have, it's first of all, establish a network of people working with this and as big reach as possible. And then our aim is to identify the gaps in soil biodiversity, microbial communities, and the question is quite complex, and there is a bit of mess in the literature. Like, depending on whom you ask, the key processes might be this or that. There might be an influence. The influence, it's in different directions. And our aim is to try to make everything as systematic as possible. We are connecting together modelers and experimentalists. And the idea would be to give a framework that can be used later on by modelers to work on possible improvements. So knowledge gap with some kind of phylogeny or some kind of systematic classification. And we are definitely looking for contributors. So if you're interested, just write me. There is quite some space to work on. To the speakers of today, 2 of them I know personally. And well, Stefano Manzoni, it's professor at Stockholm University, quite experienced in the field, I would say. And of his approach, I like a lot the simplicity. Like, he works with quite complicated stuff, but manages to be quite synthetic in his studies. With Elisa, we cooperate often. Basically, weekly we meet. And she does work on same stuff. She's been publishing really a lot on the, I would say, the big picture of terrestrial ecosystem greenhouse gases. So working on this specific topic, microbial interactions, but not only. Erik, I met just briefly once, and I'm looking forward to his presentation because it's pretty interesting. At least for me, that topic is probably the most attractive of today. And I hope we will get to know each other soon. And I think I kept it in 6 minutes, so I didn't steal so much time. Go on for the presentation, thanks.
Great. Thanks, Lorenzo. I think then it's my turn, right?
Right.
Super. Thank you very much for the very kind introduction. Please somehow wave or let me know if you cannot hear me or if there are some breaks in the connection. So I'm going to share my screen now. Just a second. This one, share. Okay. Are you seeing the screen in presentation mode?
Yes.
Yes.
Okay, perfect. All right. Well, good afternoon, everyone. My talk is going to be a bit of an introductory talk on modeling of organic matter decomposition. And I'll go a little bit through the history of how we model and we conceptualize decomposition. And I'll go a little bit more in depth on some of the challenges that we are facing. So hopefully this presentation will be a good starting point for the more, I guess, scientifically interesting presentations by Elisa and Erik. So let me see if I can, all right. So decomposition is the process that transforms organic inputs into mineralized products. Organic inputs are deposited to the soil. They follow different pathways, but they end up anyway decomposed by extracellular enzymes produced by the microbes. So in this way, microbes get access to some relatively small compounds that they can use for growth, for energy production and with emission of CO2, and also for producing more extracellular enzymes. And when microbes die, their necromass is recycled again as a substrate. Now, this circle of organic matter being decomposed, becoming microbial biomass, becoming substrate again with losses of carbon in terms of CO2, but also mineralization of nutrients, is mediated by clearly biological processes. Of course, the microbes are biological entities, but also physical processes that control somehow how much of the substrates the microbes get access to. The physical processes also control how much of the substrate is, for example, adsorbed to mineral surfaces or encased in aggregates. And there are also ecological processes in place, because not all the microbes are the same. They compete with each other. They compete with the physical processes as well. So organic matter decomposition in a nutshell is quite complicated. It involves all these processes together, and they all interact in ways that are not easy to predict. And yet, it's sometimes remarkably simple. In this plot on the right, you can see a decomposition trajectory. These are some leaves that are left to decompose in the field in a classical litter bag experiment. And as you can see, the decomposition trajectory is almost a perfect exponential through time. So this suggests that there is one main time scale that rules the whole process. So this evidence showing very simple trajectories of decomposition motivated the development of the first mathematical models of decomposition. And these models were based on the principle that the more carbon you have, the faster, not the faster the decomposition, but the higher the decomposition rate, and that at any given instant in time, a certain the decomposition rate will be given by a constant, a decay constant that you can see here, times the amount of carbon that you have in your system. And as far as I know, the first mention to such a model, at least conceptually, dates back to 1933 with the work by Salter and Green. And this formula, if you integrate it, this differential equation, if you integrate it, you get an exponential function, which is what you would need to capture this trend on the right-hand side. But what are the implications of, or I should say, the assumptions behind this formula, and therefore also the implications? Well, we assume that all the carbon is pretty much the same. It's all in one pool. It decomposes according to this proportionality constant K, or decay constant, which is fixed through time. And the hidden implication here is that the microbes are acting somehow in a miraculous way without being included in the equation. They are hidden. They are hidden in this K value. Okay, so things may appear simple, but if we look at the other data sets, maybe we start seeing some more complexity. Here you can see a decomposition trajectory over 5 decades in a bare fallow experimental site. And already you can see that the exponential function will not capture much of, it will capture the declining trend in the carbon remaining, but it will miss the initial fast decay followed by a later slower decay. So this suggests that one carbon pool, at least if we look at such long time scale, including not just litter but also mineral soil, then we will need at least 2 pools. And perhaps these pools can both decay linearly, and we can combine them to get a net effect that would capture these multiple time scales of decomposition. And this is the idea behind the classical models of carbon cycling in soils like Century and Roth C. But the concept can also be extended to a continuum of decay rates, which was explored by, for example, Joran Ågren and Ernesto Bosatta, and also more recently by others. Okay, so then this means also that perhaps organic matter decomposition is not so simple as it might look. We might need to consider different time scales with faster decay at the beginning and slower decay at the end. But overall, still conceptually not very complicated. But if we think about it for a moment, we might wonder, where are the microbes? Because in the very first slide, I kind of showed our conceptual understanding of decomposition as centered around microbial activity. And as Josh Schimmel quite nicely summarized, the microbiology, rather than being non-existent in these very simple models, is just implicit. It is buried in the equation structure of a model as kinetic constants and response functions. So the microbes are there just in a very implicit way. And implicit microbiology is better than nothing, one could argue, but it doesn't really help us figure out what's happening if the microbes change through time. For example, due to changes in land management or changes in climatic conditions. So if we want to understand how microbial changes affect the decomposition, we need to include them explicitly in the models. And we can do that through nonlinear kinetics. So instead of having decomposition just as a function of how much carbon you have, we need to include in the decomposition function also the microbial biomass. In that case, we might end up with an equation that looks like a Monod function, or we could look a little bit more in mechanistic detail and include extracellular enzymes in the decomposition function, and this would be like a Michaelis-Menten kind of equation. But we need to keep in mind that this, the theory behind these Monod or Michaelis-Menten formulations was not developed for soils. It was developed for well-mixed conditions in the lab. And this is nicely summarized by a quote here from a paper by Monod in 1949, The most important assumption is that the cultures, because of course Monod was dealing with cultural studies, should be constantly mixed, homogenized, and in equilibrium with the gas phase. And of course, we all know that soils are as far as you can get from this assumption. Soils are heterogeneous, they are non in equilibrium, definitely not homogenized. And yet, we tend to use this formalism through Monod equation or Michaelis-Menten equation at scales that are clearly that clearly include a lot of heterogeneities, and I'll get back to that a little bit later. But so now, if we include microbes explicitly in the model, are the models getting any better? And in a way, they are, but it's not so simple. So for sure, nonlinear models can capture complex dynamics, like this fluctuating behavior that was experimentally found around a root in the rhizosphere, while a linear or microbial implicit model cannot capture these fluctuations, as you can see with the dashed line. So these microbial explicit models can be useful in some contexts, at least when looking at the small-scale processes. Also, when we run them at the global scale, they allow improving a bit the predictive capacity of global scale patterns in soil organic carbon stocks. For example, the millennial model can predict 26% of the variability in soil organic carbon stocks against an 18% of the century model. Still, these figures are quite not very satisfying, so to speak. We are still far from understanding much of the variability of global carbon stocks. And adding microbes is helping, but just a little bit. The problem is that by adding microbes in our soil carbon models, we end up with a massive increase in prediction uncertainty. So changing a bit the model structure will basically lead to diverging predictions of carbon stocks, especially into the future, as Bill Wieder showed very nicely in a paper in Global Change Biology in 2018. And part of the reason we have such an increase in prediction uncertainty is that the nonlinear microbial explicit models tend to have strange behaviors that we don't fully understand. Take this example here at the bottom of the slide on the right. Again, a little decomposition data. A microbial implicit model does a really good job in capturing the trend. If we try to fit a microbial explicit model, we see that the model starts doing okay, then decomposition becomes way too fast, and then it gets stuck somehow. So that's a result of the nonlinearities, and this is just one example. Erik will show some more in the following talk. So we are a little bit stuck here because we would like to add microbes to increase realism and the mechanistic detail in our models, but we face some challenges. And this is a very biased perspective of what some of the challenges are. There are other challenges that we can also add, but at least in my opinion, some that are interesting to look at are the unexpected behaviors of the nonlinear microbial explicit models, and Erik will talk about that, the scale mismatch issue, and whether we need to include microbial biomass or what other features of the microbial community in our models, and Elisa's talk will be about that. But now let me talk a little bit more about the scale mismatch issue and whether we want to include microbial biomass or some other features of the microbial community in the models. So scale mismatch. To understand the issue, why it matters to think about scales, we can maybe consider this very simple example. Think of the soil as composed of two patches. One patch identified with the subscript 1 and one patch with subscript 2. And within each patch, conditions are perfectly well mixed. So we can apply theory even from lab experiments, Monod equation, for example, or Michaelis-Menten equation. But we don't know what's the behavior of the whole system, so the combination of these two patches. So what we would like to know is the mean process rate, which I indicate with F with an overbar, using only information about the mean driver of the process, X bar, because that's what we really care about. We care about rates and stocks at a relatively large scale, and within this large scale we can have smaller patches, okay? So what we are tempted to do is to look at the mean driver, for example, the mean organic carbon content, and calculate the process rate at the mean carbon content according to the same formula that we would use at a small scale, which is depicted in this figure in blue. Think of this as some Monod function, for example. Oftentimes, we just take the mean soil organic carbon and calculate the corresponding rate using the Monod function. But that's actually incorrect, because the mean rate is the mean or the average between the rate in the patch 1 and the rate at patch 2, which is here, the green point, the green circle that I highlighted here. So this is the correct average rate in the system, but we often tend to use this one. So we end up with biased estimates. So in soils, we can assume that conditions are relatively well mixed when we are at scales lower than 100 microns or even less if the soil is not too moist. At these very tiny scales, perhaps we can apply Monod equation or Michaelis-Menten equation. But what will the kinetics look like when we average across all these patches in the soil? Can we actually come up with some kinetic law linking the mean rate to the mean driver at the large scale we are interested in? And we can do that proceeding formally using the so-called scale transition theory. Scale transition theory starts from an assumption on the small scale kinetics, in this case of decomposition, which we can call F of C, where C is the carbon content. It also requires some information on the heterogeneity of the soil at the small scale, for example, the standard deviation of the organic carbon. And then we proceed, mathematically, we proceed by special averaging via Taylor expansion around the mean. So the mean rate of decomposition in a heterogeneous system, according to this theory, I'm not skipping all the details and giving you the result, is going to be equal to the rate calculated at the mean soil organic carbon content, which we call mean field approximation, and it works perfectly well for homogeneous conditions, plus correction terms that account for the heterogeneity. And these correction terms involve sensitivities of decomposition with respect to the carbon content and information about the spatial heterogeneity, this variance of carbon. So to give you an example, let's assume that at the small scale, decomposition is controlled by a multiplicative model, where the more substrates, CS, you have, the faster the decomposition, and the more microbial biomass you have, the faster the decomposition, in a multiplicative fashion. Now, if we apply scale transition theory with this model for the small scale, we end up with a large scale model that contains a first term, which is basically equal to the small scale model evaluated at the mean carbon content and mean biomass content, plus a correction term that accounts for the heterogeneities. And if for this specific multiplicative decomposition, this is equivalent to it's proportional to the covariance, the spatial covariance of substrates and microbes. So if the microbes are not co-located with the substrates, just to give you an example, the covariance will be negative. This means also that the large scale flux will be less than the mean field approximation. So if we apply this formula, the small scale formula at the large scale without correction, we will largely overestimate decomposition. And this is what you can see here in this result figures, where the homogeneous soil, where there are no heterogeneities, will behave in a very clear way with a strong decomposition leading to complete consumption of the substrate. While in the heterogeneous soil, we actually have a slowing down of decomposition, and eventually some carbon will remain in the system because of this inhibition effect due to the heterogeneities themselves. Now, interestingly, when we apply this scale transition, the kinetics of decomposition, which are now represented by this large scale function, will look quite different compared to the kinetics in the small scale. So here you can see some examples, but there are with other decomposition models, we will also have similarly different kinetics. So the bottom line here is that when we look at the large scale and when there is a significant heterogeneity in your system and you want to apply nonlinear models, at the large scale, the kinetics will look very different from what you would assume to hold at a small scale. Now, the second part and last part of this talk is more about what should we add in decomposition models to capture the effect of microbial communities. Should we include microbial biomass like I've shown in this example here with a multiplicative model, or should we consider other features of the microbial community? Well, I also already talked about extracellular enzymes, but how about richness or functional diversity of the community? Could they be good predictors of decomposition to be included in a decomposition function? Well, maybe so. Theory predicts that diversity should be positively correlated to decomposition because more diverse microbial communities will occupy more niches in the soil and therefore will be able to use substrates more effectively. And indeed, if we look at the lab data, we can see a clear proportionality between the functions like respiration or microbial growth and richness in the system. This is a log-log plot, and this relation therefore is a line in log-log plot is like a power law in a linear plot with an exponent lower than 1. So this means that the effect of richness kind of tapers off as we move up towards more and more diverse systems. But also you can look at the values on the x-axis here. They are fairly low compared to the species or operational taxonomic units that you would find in reality, because these are data from extremely simplified communities in the lab. But to study these diversity effects, Swamini Kurana, a postdoc in HoliSoils in my lab, built a model where she could actually manipulate diversity in the model in two ways. She could change the number of microbial groups that consume different substrates, but she could also change the traits of such groups. She was extracting the values for the model parameters from random functions, and in this way, by changing the variance of such functions, she could also manipulate the functional diversity in the microbial community in her computer simulations. And what she found is that three features are particularly important to understand and predict decomposition patterns in her model. One is the amount of carbon present. The dark lines here are all corresponding to a system with more carbon, and the light brown or orange lines correspond to a system with less carbon. So if you have more carbon, this will fuel microbial growth and therefore speed up even more decomposition. So there is a reinforcing positive feedback. Now, regarding diversity, she found that increasing the functional diversity has also an effect. Like these dotted lines are all higher compared to the solid lines, and the dotted lines correspond to high functional diversity in the microbial community. So this means that we can parameterize the decay constant of even a very simple linear model as a function of diversity and carbon content. You will end up, of course, with a non-linear model because you have carbon also here in the definition of K. But what I want to focus on is the diversity part. So with higher diversity, we will have a faster decomposition with an exponent beta. So it's again a power law. And this beta F, according to Suomini's model simulations, should be around 0.1. And here it's this diamond in this plot, and you can compare it with the data, the box plot, and you see that it's not far from about 0.15 that we estimated from the data. So what does it mean? It means that according to Suomini's simulations, diversity should matter. It should matter particularly when the microbial community has low diversity. So a small increase in diversity in a low diversity community has a much larger effect than an increase in diversity in a highly diverse community. And also, it's interesting to notice that the model results are consistent with the data. So, take-home message, microbes drive carbon and nutrient cycling, and we certainly understand how they grow, at least in idealized conditions, so we can model their growth in such conditions, but it's challenging to implement knowledge coming from idealized situations in operational carbon and nutrient cycling models that are used at much larger scales. And this is partly because nonlinear models that include microbes exhibit complex dynamics. It's challenging also because the decomposition kinetics in theory actually change with scale. And also, we should, according to Suomini's results, we should keep into account also microbial diversity or functional properties of the microbial community, which actually also change through time. So it's not really easy to just add a diversity index in our models. So I think the next steps to progress in our understanding and modeling of decomposition are to, first of all, understanding why microbial communities change, therefore affecting decomposition. And this is partly work done by Elsa Abs and Erik himself and others, looking at how ecoevolutionary processes change the way microbes behave and therefore would require a change in the model parameters through time. But also, and perhaps this is the elephant in the room, we should better integrate physics and biology in soils. And there are really excellent conceptual work by and also quantitative work by Philippe Bavey, but also Jin-Young Tang and others, really trying to see how physical constraints affect the decomposition beyond the biology and ecology. That was the focus of my presentation. So there is a lot more, but in 20 minutes, that's all I can say.
Thanks, Stefano. And it was a very good introduction to the problem, actually. We have already a pair of questions, but I would leave them for the end because I think they could be also corally answered. I would say we go straight to Elisa's presentation and then we see if we have time to answer them.
It sounds good to me.
And
otherwise, if I need at some point to leave, I'll try to answer questions also via email if they are for me. Okay.
Yeah, to everyone, I remind you, please ask your questions. We will find a way to answer them. I leave the floor to Elisa.
Okay. Okay, can you see my presentation? I guess so. Okay.
Yeah.
So I will continue this webinar after this nice introduction from Stefano to present a work that we did together with Stefano, Lorenzo, and other colleagues in HoliSoils, where we try to validate or understand from empirical observations if microbial diversity integration into soil carbon models actually improve the predictions of carbon decomposition. So the background context of this work was that within HoliSoils, we wanted to understand how natural disturbances and forest management affects greenhouse gas emissions. And we know that these disturbances have effects on microbial communities. In particular, often disturbances would reduce the microbial diversity. And what is still unclear and what we wanted to better understand with this work was how the microbial diversity impacted in turn CO2 emissions and other greenhouse gases. And so the answer that we wanted to, the question that we wanted to answer in this work was, how does microbial diversity affect carbon and nitrogen mineralization? So the work that Stefano has shown earlier from Purana theoretically demonstrated that we should expect a positive relationship between microbial diversity and carbon decomposition. So here, again, it's a power law. So it's a logarithmic graph, but what it's showing basically is that microbial diversity and carbon decomposition have a positive power law relationship. And so what we wanted to do with this work was to test whether we actually observe this positive relationship in empirical data. And in particular, if integrating microbial diversity into a carbon and nitrogen model would improve the predictions of the mineralization rates. Okay, so to do that, we used a very simple approach. So if you remember the carbon model that Stefano showed earlier, this is something very similar to it. So we have that the variation of carbon over time is equal to the input of carbon from net primary production minus the outputs of carbon from the soil, which depend on the amount of carbon in the soil and a certain decay constant K. And this decay constant K is multiplied by this C parameter, which is a function of temperature and precipitation, usually in this kind of models. So basically you have an effect of climate that modifies the decomposition rates of carbon. And so the idea here was to integrate a function of microbial diversity, F, which would also modulate the decomposition rate of carbon according to this exponential function, where we have F, that is our microbial diversity index, and beta, which is a coefficient that we would calibrate on data, and that would tell us the direction of the relationship between microbial diversity and decomposition. So if beta is positive, we would expect, we would have a positive relationship, which is what we would expect according to the theoretical background that we have. Whereas if beta is negative, then we would have a negative effect of diversity on the decomposition. And finally, if beta is 0, it means that there is no effect of diversity on the decomposition rate. And so we integrated this modeling framework. So we use this modeling framework on a set of data that was collected by some Spanish colleagues a few years ago. So one of our colleagues is Jorge Curiel, who was also on HoliSoils. And so they collected soil samples around Spain in about 20 sites that were Olmec forest sites. And they incubated around 180 soil samples and measured carbon and nitrogen mineralization, and also microbial parameters like microbial biomass. And also they sequenced DNA sequencing to measure the diversity of the microbial communities. So the diversity was based on taxonomic identification, and this taxonomic data was used to estimate different diversity indices for different microbial groups. So we estimated richness, Shannon, and inverse Simpson for bacteria, fungi, a combination of bacteria and fungi, and two different fungal groups. And we tested the model with all these indices. And what I will show today is just the results with the richness of bacteria and fungi combined. So this is our F in the exponential function. And we had very similar results with all the indices. So the story doesn't change much. So what we did basically was that we started from a very simple model, model 0, where we had no effect of biomass, which is represented here by this gamma exponent. So gamma was 0, and microbial diversity also was set to 0, so beta here is 0. And then we started adding some processes and test how it affected model performance. And so we have model 1, where we integrate the effect of microbial biomass, but no effect of diversity. Then model 2, where we have diversity, but no biomass. And finally, a full model with both diversity and biomass effects. So what we found here, I am showing the plot of predicted versus observed respiration rates. So I'm just showing results for the carbon decomposition. If we compare results from model 0 and model 1, you can see that the performance of the model here with the R² is increased when we integrate biomass. Instead, when we integrated diversity, we had we did not improve the predictions of the model. And same when we already have biomass, the integration of diversity does not improve the predictions much. And so what we concluded was that integrating microbial biomass improved the prediction of carbon respiration. And if we consider the Bayesian index criterion, which penalizes models that have more parameters, so to benefit or prioritize simplicity in the models, then we would select the model with only biomass because it improved predictions without adding too much complexity and free parameters. So concerning the diversity effect, so if you remember the beta parameter that I showed earlier, we found that the distribution of the parameter was very close to 0. So it means that diversity had a very small effect, as I said earlier. And also the sign of beta was negative, which means that contrary to what we were expecting, the effect of diversity was also negative. So it was small, but still negative. And instead, the gamma parameter, that is the biomass exponent, was very close to 1, which indicated an almost linear effect of microbial biomass on the decomposition rates. And then we also had this the decay constant and the carbon use efficiency parameters that we also calibrated with the data, and that were well constrained. So This study had a few limitations. First, we considered microbial diversity based on taxonomic diversity, which might not be the best way to reflect the effect of microbial functional diversity and its effect on ecosystem functions. Also, we considered carbon use efficiency and the CN ratio of the microbial communities as being constant, so the same value throughout all the sites. But we know that these parameters may vary spatially, especially depending on the communities that are present and the substrate also that is in the soil. So what we are trying to do now with Lorenzo is to test another approach to go a bit further in trying to extract some information from the taxonomic data. And so this is a hybrid modeling approach that with other colleagues also from the Max Planck Institute in Jena, we have tested. So basically, the idea is that we, instead of calculating microbial diversity indices from the taxonomic data, we feed this data to neural networks that will predict the variation of the carbon to nitrogen ratio and the carbon use efficiency of the microbial communities in space, and then we would inject these neural networks in the model to predict the carbon and nitrogen mineralization rates. So this is still work in progress. For now, we hoped this approach could tell us more. It's a very nice approach, but our preliminary results seem to suggest that there is really not much that the diversity, not much more that diversity can explain in this data. And so here I'm just showing different diversity components here because we kind of synthesize the taxonomic data into with principal component analysis into a lower number of components. And here we are showing that basically model performance does not depend much on the communities that are there. So this is all. Thank you very much for your attention.
Thanks, Elisa. Adding a small thing on the last step, just to mention that we are using exactly the same data set of the first study that Elisa presented. Different data sets might give different answers. We do have time for taking the questions, but I think there would be questions directed to all the panelists because they are general enough and they connect pretty well. Like one of the questions is about functional diversity versus population diversity. And the other, which I think connects very well, it's about arbuscular mycorrhizal fungi, how to model them. And yeah, I guess that's very big important of functional diversity in that case. I think it was originally addressed to Stefano, but does any of you have ideas about how to answer this? Elisa, maybe on the functional diversity versus just diversity indicators like species diversity, maybe you can argue a bit.
But yeah, I think it was, as I said, one of the main limitations of this study. So we should expect that, so there might be some redundancy in the soil of functions that different communities can provide, and so taxonomic diversity doesn't really reflect the functions that communities can provide. And yeah, for sure, it's something to try out that probably could reflect more. Yeah. the effect of diversity in the soil.
I would have given same answer, actually. We've been hitting this species diversity. We had a lot of hopes about it, but it seems that in soil there is so much redundancy that when you compress the information that way, it ends up not telling really so much, because there is anyway an enormous amount of species diversity. It would work differently if we were talking about less diverse ecological communities like plants, macrofauna, mesofauna, maybe. The mycorrhiza one, I don't have direct experience with it. We did find quite some importance of fungal communities so far, but I never worked personally with data with mycorrhizal fungi.
Perhaps I can comment a little bit about models that include mycorrhizal fungi, and there are now several. Maybe they can also be used as predictive models, though I'm not entirely sure. The first question was about process-based models or statistical models. In any case, there are models like Mimics Plus that include now ectomycorrhizae and arbuscular mycorrhizae. But this is just an example. There are several other models that do include mycorrhizae. It's still quite tricky to parameterize them, though, because yeah, we can always add pools to our models, right? But with each pool, especially microbial pools, we are adding at least 4 or 5 parameters, which are generally largely unconstrained. So conceptually, it makes sense because we are adding our knowledge into a model, but then can we actually use it and can to get some robust answers? That I'm not entirely sure. But again, I added a link, a couple of links in the question and answer discussion or chat. Also answering the other question about parameterization of K in the decomposition, whether to worry about population diversity or functional diversity, and some thoughts, but these are just my ideas.
From a purely procedural point of view, actually, I would suggest if you have those data, it would be nice to fit a simple model, like just an exponential model, and then explore the residuals and the correlation of with arbuscular mycorrhizal fungi. If there is a pattern in the residuals, it would already offer some information. Like if you can predict the residuals with the AMF data, that means that, okay, we should do something about it.
Although, I mean, I'm not sure arbuscular mycorrhizae contribute much to decomposition per se. Let's see. Yeah, I mean, I know the question was not really about decomposition, it was more in general. Sorry, forget what I'm saying. I'm telling nonsense.
The question there would be, do we work with soil respiration or total respiration? Like if there is only the heterotrophic or if there is everything, it will change quite a lot. Oh, another question. Oh, no, not a question. Thanks for everything. Thanks for the comment. Maybe we could go on Erik talk. We avoid silences.
Sure. In the screen. That's not my presentation. This one is. So you can see my slides. If I don't hear anything else, then I'll continue. Perfect. So yeah, thank you very much for this entire webinar, and thanks Stefano for laying the foundation basically for everything that's gonna pop up in some form in this talk. So I mean, because this is a work that we did together with Stefano and also Samia and Celine, and that was a bit motivated by us working with microbial explicit models, and finding that they become unstable when we do simulations. We struggle a bit with understanding where this comes from. So this is basically why we did the study, to try to really understand and get to the bottom of when we can get instability in those microbial explicit models. And now stability sounds awfully technical, and it's late, like we're an hour in after presentation. So I try to keep the math out of it as much as possible and try to focus a bit more on the processes. But we've published this now also as a paper. So if you're really interested in the mathematical formulations, I'll have a DOI at the end of the presentation, so you can look a bit more into the fun analytical mathy part of the side of this work. So Stefano already introduced a bit how we conceptualize processes in soil, and that's where I start off as well, just to give a very brief recap of those processes. So with microbial explicit models, basically we want to describe explicitly how microbes decompose soil organic carbon. So those models always represent a microbial component and then some organic carbon content or compound. Most of the time we assume that microbes cannot directly consume that substrate, but they probably need to produce some extracellular enzymes, which then depolymerize a more complex substrate, and from that produce something that microbes can actually consume, which is low molecular weight organic compounds, or here indicated as DOC, as dissolved organic carbon. And this is then some more monomeric substrates that can go into the solution, diffuse to the microbes, and then also be taken up by them and being consumed. And here this P basically represents the depolymerization rate that is ongoing, that's catalyzed by the enzymes and produces the DOC, and the U is the uptake rate, so the consumption of those monomeric substrates by the microbes. The microbes use that in different ways. First of all, they drive their metabolism, so they respire some of it to generate energy. They also use it to grow, to produce more biomass, and of course also more enzymes. And then they also die, and some of that microbial biomass, necromass, is recycled into the soil organic carbon pool. And basically, conceptually, this is what also then guides the carbon fate. So we have a balance between how much is respired of microbial carbon cycling, again, to CO2, to the atmosphere, and how much is stabilized and remains in the soil. Now, from this very simple perspective of those turnover processes of soil organic carbon in the soil, we can also build very simple models. And this is here some sort of archetypical model. That means it's in some form the backbone of many other microbial explicit models. Specifically here, this is the based on the Ellison-Wallenstein-Bradford model. And this represents exactly those processes that I've just described. So the depolymerization of SOC, which is here catalyzed by enzymes, which produces DOC, so some substrate that microbes can actually consume, the nonlinear uptake of this substrate by microbes, and then how microbes produce enzymes and the respective decay fluxes and so on. So with those models, they have basically different mathematical properties that are inherent to a specific model structure that come with it. And one that is very important is the stability of those models. So stability here means basically how a system that is at an equilibrium reacts to a small perturbation. So if we have a system and we perturb it just by a tiny bit, how does it react after that perturbation? And usually what is most suitable for us and what we want to models to do is that they react stably, so that they're stable. That means they return to the initial equilibrium. So they're stable to that perturbation. And that can even either happen that they directly go back to the equilibrium, as it's shown in the blue line here. It could also be that there's some oscillation first, so maybe they overshoot their initial equilibrium, but then with dampened oscillations go back and reach their initial state again. And all of those behaviors are basically considered and classified as stable behavior, because we end up at the same equilibrium after such a small perturbation again. However, what we can also observe is that those models can be unstable. So that means after a small, ever so tiny perturbation of the system, it would not return to its original state, but it would go somewhere else. And what we observe for the microbial explicit models, that usually means that the microbial biomass dies out. So the system sort of crashes. exactly, those are those two classifications. Now, what does that mean for models? So Stefano already introduced that. And if we look at the scales at which those models are conceived, so really small scales, we could basically imagine all of those behaviors to be realistic, right? Stefano already showed this oscillations that we can observe in microbial systems, but we could also imagine that those systems, or that at those scales, the microbial community suddenly dies out because maybe there is not enough input flux coming to a microsite anymore, so the microbial community might die. So here we don't really have any problems. That's within the realistic behavior that we would expect. However, again, the scale issue, if we want to use those models, and this is usually what is done, that we take this sort of models and try to apply them at a global scale to make predictions of soil organic carbon dynamics on global scales, then we run into some issues because then we don't observe anymore that on a square kilometer scale, all of the microbial biomass suddenly dies out, which is what would happen if the system is unstable. Also, we have some other technical issues because then our spin-up simulations don't work, and we get predictions that are, if we base our predictions on steady-state analyses, they might then be unrealistic in those scenarios. So if we want to move ahead and use those models at larger scales, we need to understand what causes these instabilities in those models and also how we can avoid them. And this is what we're doing in this work. So we were taking this model that I introduced previously, and we use different methods. Here we looked at analytical, descriptive, and numerical methods. But as I said, I focus mostly here on the descriptive part. So what's going on in those models? And to really understand what's happening, we can just basically take a tour through the model and see what happens if we slightly perturb it at some point. And we start here with the DOC. So basically, we could imagine that for some reason, we're in an equilibrium and there's a slight perturbation of the DOC concentration, maybe some rain event, and we end up with less DOC. So what happens if we propagate this decline in DOC throughout the system? Well, in the model that we have, we describe a lot of causal relations, right? So this is indicated here with those green arrows, and the plus is a positive causal relation. So what those models describe is that if we have a decline in the DOC concentration, we'll also see a decline in the uptake rate because they're directly coupled. And if we have a decline in the uptake rate, we'll also have a decline in the microbial biomass concentration. So here we have a positive causal relation, right? A decline in DOC leads to a change in the same direction in the microbial biomass pool. Now we can continue with this causal chain and look what happens further. So if we have less microbes, we'll also have a reduced enzyme production, and we'll have a reduced enzyme concentration in the system because less microbes produce less enzymes. Now, if we have less enzymes, that will slow down the depolymerization rate because we suddenly have less catalysts. And if we have less depolymerization ongoing because we have less catalysts, that will mean that, again, we'll have less substrate available. So what we've created here with those models is a positive feedback loop. So an initial decrease in the DOC concentration will, by the means of the causal relations that we have in this model, cause a further decline in the DOC substrate concentration. And now, because this is a reinforcing feedback loop, what we get is that this can spin out of control, and eventually the microbial biomass could die out. So what we observe is that this is an element that can lead to instability in the system. However, it's not always that those models are unstable, so there needs to be a balancing part as well, and that's what we find if we look at the other side of the model, which is the SOC part. So basically here we have a balancing component in the system, because if we look again at the depolymerization rate, if we decrease it, that means we'll get actually an accumulation of the SOC, because we remove less of the substrate that will accumulate. So we have a negative causal relation. That means an accumulation, which will in turn lead to an increase in the depolymerization rate again. So because of this balancing feedback loop, we get basically a different sign introduced into the system. So suddenly the depolymerization rate increases again. And this again can propagate throughout this feedback loop. So here basically we have a balance between those two feedback loops, the reinforcing and the balancing one, and stability that very much depends on how they're weighted in the model that we have. So analytically, this is also, if we do a thorough analytical analysis of that model, this is exactly what we find. So basically we can find a condition that describes when the system can become unstable, and that very much, or that exclusively depends on the quantitative. Here it's expressed in sensitivities, the sensitivity of the depolymerization rate to this change in the SOC concentration. This is the quantitative, like quantifying this balancing feedback. And on the other hand, the sensitivity of the depolymerization rate to a change in enzymes, that it's quantifying this positive reinforcing feedback loop on this side. So this is how instability comes into those systems. But what's really the cause of this? Why do we observe that? So we tried to go a bit deeper into that, and we also looked into other studies in the literature of similar systems. And what we have observed, what is similar between all of those different models, is that we get this instability if we have the growth substrate, so something that an entity needs to consume, if the replenishment of it, so this is this depolymerization rate, right? So the DOC is replenished through the depolymerization. So if this replenishment is directly through a positive feedback loop or any other direct relation linked to the abundance of the growth substrate, that's when we observe that those models can become unstable. So we also tested that by taking out the DOC pool. So some models actually do the simplified assumption. So they just assume that the depolymerization rate is actually equal to the uptake rate. So everything that is broken down by enzymes is directly consumed by microbes. So in those models, we basically skip this representation of a DOC pool. So the growth substrate is the SOC. It's outside of this positive feedback loop. It's replenished through litter fall and not dependent on anything else in the system. And for those models, we always find that they're always stable. So this indicates further that actually the problem is displacement of the growth substrate in this positive feedback loop. Now, what does that mean for the modeled predictions that we get? When would we observe or expect to observe instability? So if we run the model with here very external conditions, so we keep all the parameterization as it's usually used in global scale simulations, and very on the x-axis, the OC, the organic carbon input rate. So how much substrate is coming into the system, and on the y-axis, how much is leaving. So how much of the DOC is leached. So how much is lost due to abiotic processes. What we find is that we get less and less stable behavior of the system as we go to less favorable conditions. So if there's less substrate available and or more of the substrate is lost. And this is also in accordance with other models that find that, for instance, in high latitude areas, usually oscillations in models are also extremely high, which is an indicator that the models there are not as stable as they're in other places. Alternatively, we could also look at how different parameters in the model influence stability. And here we focus on some microbial characteristics. So here we see on the x-axis, it's the microbial substrate use efficiency, and how that affects stability in the system. And here on the y-axis, we have the dampening coefficient. So that basically indicates whether a system is stable or not, based on the sign of this dampening coefficient. So if it's positive, if we're above this 0 line, then the system is stable. If we're in the negative, it's unstable. And what we see from this analysis is that if we're in very favorable conditions where there is a lot of food and very little is lost, microbes basically can do whatever they want, right? So this microbial substrate use efficiency, which is similar to the carbon use efficiency, does not really affect the stability behavior. If there is a lot of food, microbes can be as inefficient as they like, and they would not die out because the system becomes unstable. However, as we go into less favorable conditions, so here the yellow and then the red line, we see that there's actually a threshold that moves upwards, right? So as conditions become less favorable, as there's less food available, microbes need to become more efficient in using that food in order to be able to sustain stable population under those conditions. And this is actually, I think, a quite interesting conclusion that we get from this analysis. And it's a quite basic one as well, because it's one of the basic principles of ecology, which is that microbes have to be well adapted to the environment they inhabit, right? You can't be extremely inefficient if there's very little substrate available for you. Right, so what's the solution? Well, already pointed some of them out. So first of all, what we could do to avoid instability in those models is to create them differently in their structure by excluding this sort of feedback coupling. So taking out an explicit representation of the DOC, for instance. Alternatively, what I didn't explore now is to have biomass independent uptake kinetics. That would also work. But all of those steps basically would lead to a simplification of the system. And then it's always the question whether this is justified, whether this is still representative of our conceptual process understanding. Alternatively, what I've just discussed is that we could also include other ecological principles into the model. So for instance, that explicitly represent that microbes do adapt to changing environmental conditions. And thereby we would actually increase model realism, while at the same time also avoiding instability behavior. So that's it from my side. I hope I sort of kept the time. Thank you very much. If you have any questions, want to contact me. And as I said, the paper is published, so you can find the DOI here. Thanks.
Okay, thanks, Erik. We do have a lot of questions now. Some have been already answered. Elisa is taking care of one that I'm beating my tongue on quality. Nobuhiko has one open question, but I have to read it, sorry. I read it aloud. Thank you for the very interesting presentation. I was particularly intrigued by your discussion on how microbial diversity is represented in the composition models. Beyond the home field advantage in litter decomposition, local matching has also been observed in contexts such as rhizosphere communities, litter mixing effects, or symbiosis. Do you think such forms of local adaptation should be explicitly considered in the composition models? Which is pretty interesting. I remember your talk, Nobuhiko, about the home field advantage. Anyone wants to take this? My guess is that yes, they should be, but then we are still a bit behind. Go, Stefano, sorry.
No, go ahead, Lorenzo.
I mean, we are still discussing on how to include diversity in itself. So I don't know how to put in local adaptations of communities.
So it's in my view, it could be one of those things that are somehow implicit in the models, right? I mean, it depends also on what model you are using, what do you want to use it for. But if you calibrate your model on some specific data sets, somehow these effects will be included implicitly in the parameter values. So of course, then the challenge is how to model these effects if you are really trying to yeah, right. So if you do like a transplant experiment, or if you take some inoculum from one place, you put it somewhere else, and you want to model those effects, then it's really challenging. Because then you need to include the mechanisms in the model. So I guess stepping back a little bit, do you need to model these effects for some practical applications, or is it more like a theoretical question that you are exploring?
Would solve it as usual with like putting more degrees of freedom, testing a model with different degrees of freedom without assuming any function of this adaptation would be yet another mechanism that would scale the hypothetical mechanism that we are putting in for the microbial diversity, or for some other mechanism to take care of communities. And then if it was me, I would just test this simpler model, which is already quite complex, and see if a more advanced model has if there is some variance left to explain because of these adaptation effects. And again, the question is, do we really need it or not? Which is pretty, I think it's unanswered.
Maybe if I can add something, it just maybe depends also on the what is your objective. Maybe if you want to predict long-term carbon dynamics, you don't really need such resolution in the model. But if you want to understand how things work, then yes, it can be a way, if you have data to constrain your new parameters, to like constrain a bit more the different processes and understand how it works. But it really usually depends on the goal of your model.
And Elisa, did you answer the question about quality? I'm trying.
Yes, I tried to. I put some references of. There are some models already that integrate particular organic carbon and mineral associated. So there are some few examples already, and I think if you want to, again, if you want to predict the long-term dynamics, then you probably need, as Stefano said in his presentation, multiple models that have different decomposition rates. I think, yes, having like measurable pools is always useful because you can test your hypothesis. But yeah, the problem I think with the POM-MAUM framework is really like global data sets usually have different methods for measuring the pool, the soil fractions, and that complicates a bit the validation of the models.
As a hardliner there, I would also argue that in a model like MEMS, you are also forcing some assumptions. You are forcing some other assumptions on the pools that are not necessarily, like there might be some missing processes. You assume that there is this mineral association that are having that effect, and that's how you describe quality. But there might be something else unaccounted for, like physical protection, especially explicit effects on the microscale that your model would not account for, and then you would have the quality of your MAUM pool might not be exactly the quality that you find in the field. It's like it's the same problem than assuming everything one single pool. Then you go a bit more complicated, which we know that it doesn't represent exactly quality. If you decompose it a bit, you can still hit the same problems. And then I would say that it depends on the data that you have. If you have data to isolate them and you have covariates that respond to those specific pools, then it's worth it. But here I have my own bias on this. I always stay away from physical definitions of pools. It's enough to use for, okay, yeah, that was one that. It's enough to use 4 years carbon data to predict long-term dynamic. I'd say it depends on the model assumptions that you're using, actually.
But in 4 years, you will probably see patterns related mostly to the turnover of the particulate fraction of the litter, but maybe not so much about the more stable pools that turn over much slower.
If you are using, if you're not calibrating your model just on this data, but for example, you are taking a model with also slower pools and integrate the previous knowledge from the previous calibrations concerning only those pools, then I guess, yes, you can predict with 4 years, but just because you're extrapolating from other data sets. The question was using a single pool model and calibrating it on this 4 years data. If that is the question, I guess the answer is no. Or the answer depends on the time scale where you want to predict also. which connects with Stefano, the first slides that Stefano showed. Maybe we managed to take care of all the discussion even a bit earlier.
I think maybe I can add one general comment. I think at least regarding Erik and myself, I guess the models we develop and use, they are mostly for understanding processes rather than predicting accurately something. And this is an important distinction to make because sometimes our models are a little bit unbalanced because we are focusing on purpose, on specific aspects that we are interested in. Maybe an ecological question or I don't know, a question on how to use a specific type of data. I mean, many of these questions that are in the chat are extremely relevant, but you might want to ask maybe other modelers who are more interested in developing predictive models. Well, Elisa, of course, is here, and you do have a lot of experience in that direction. But just keep in mind when we answer your questions that the perspectives change a lot between, let's say, Erik and myself and Elisa. We already have quite different perspectives. And then comes Lorenzo with his data-driven thinking, which is even more different. So when you read our answers, so when you contact us via email, keep in mind we think a little bit differently about these problems.
Okay, I would say if there is no other questions, there is one open question. There are questions coming. About the models working in the tropics or not. I have some experience with the ICBM is the model that I've been using for 10 years before moving to other models. I did test it, I think end of 90s. They kind of work because it depends on what are the challenges that the model will find. Like the temperature scaling, I think it's pretty well tested. So different temperatures shouldn't be particularly problematic for most models. Particularly if you are not interested in very fine predictions, like if you are fine with 80–90% of the predicted variance, I would say that I would use them. What might be problems there? Actually, you can have enzyme denaturation if you are at very, very high temperature, 35 degrees, but I don't think you find it most often in soils. The moisture scaling, how would it work? It's not exactly my field, the moisture part. What might be the limits of moisture scaling calibrated on our soils up here? In the Nordics, moisture scaling is rarely a problem because soils are often in the optimal range, actually. It might be that something unexpected happens when the soil is very dry. Would be worth testing it.
I mean, my first thought thinking about the tropics is that the soils are very different and they are oftentimes older. I mean, not mixed up by glaciers or so it's possible that you actually have a more significant effect of mineral interactions that maybe is not so strong in some other soils in the temperate areas. But I don't know. I haven't really thought about it. If you look at just litter, the main rules are the same in warm and cold temperatures. More or less, I guess you can predict the decomposition in a similar way or with a similar model structure. But I'm thinking about litter bag data, which are the most common. Of course, in the tropics, you have, of course, much more faunal activity, which in reality contributes to decomposition more than in temperate areas. and maybe that is not accounted for in your model. So it depends very much on what you are really thinking about this temperate tropical comparison. Is it litter? Is it soil? What type of soil? It's hard to give answers, clear answers.
Those clays should be less reactive than the clays that we have here. Like older clays tend to have less cation exchange capacity. But this is just memories from my university years. So they might be being not as active. The mineral protection might be a bit less.
I think also maybe for the model parameterization, usually like models are empirically calibrated on data, which are mainly from temperate soils. So there is probably a bias in the, for example, in the decomposition rates of the pools, because in tropical soils, for example, like decomposition is more rapid. So there is some work, for example, on the Yasso model for forests that was done by Guandeu in Benin, where they calibrated Yasso on litter bag experiments in tropical soils. So there is some work to validate or calibrate the models in also non-temperate sites. So there's like probably issues also in the parameterization. So if you use a model, apply a model in tropical soils, then you should recalibrate it.
Okay, sorry. Trying to follow the question. So, Erik, it says that you are going to answer the question live.
I can try. Yeah, so the question, maybe I should read that out, is a bit about how to model the reaction of microbes to perturbation or microbial decomposition to perturbation, for example, to extreme temperature during a heat wave or drought in a peatland soil. Do you let define the model the correlation between parameters, or do you base that in field lab experiments? From Andres. Yeah, thanks for the question. I think for the analyses that we did, at least now trying to connect that to that, their perturbation is maybe not what you would think of experiment in any experimental or ecosystem process context, but I mean there it's about really tiny mathematical perturbations. So basically, just because I mean maybe your parameter is off by 0 point something or so, or there's just some numerical fluctuations. Just to give the definition of what the perturbation, what kind of perturbation analyses we're doing with those mathematical analyses. But I think if you want to apply that more with in terms of like an ecosystem process, I think then it becomes relevant really to have the specific understanding of the specific process. For instance, like nonlinear reactions to temperature changes, as for instance, Lorenzo indicated that at some point enzymes just denaturate. And I think one of the major challenges, I think, in integrating processes then is maybe to look at different microbial reactions in terms of stress evasion and stress resistance. So whether they try to combat a heat stress or so, or for instance, drought stress by producing osmolytes, or whether they turn dormant in order to evade it. So then we're a bit back to the question that maybe Stefano raised a bit, whether we want to really go in detail into the specific processes and want to understand them, or maybe we want a more empirical understanding of the consequences of those effects. I think that would be my understanding.
Yeah, the recording will be shared. This is the question, not for us, but for the meeting organizer, meeting host. I guess it will be shared.
Yes, the webinar will be shared through the HoliSoils project website, and also it will be available in the YouTube channel of the European Forest Institute account. So once the webinar ends, we will also ensure that the recording is available, and we will also send an email to all the registered attendees with the link.
I guess our time is over now. So many thanks to particularly the panelists for the very interesting discussion and talks, and also to the public for the engaging discussions. And see you everyone soon, I hope.
If I can add something, Lorenzo, before ending the webinar, I have included in the chat the information of the HoliSoils Webinar Series that it will be run in September and October, so you can find all the sessions. I shared the link in the chat, so you can see in holisoils.eu/webinars all the sessions that we are organizing in September and October, and also there will be sessions in November that we will soon confirm, so you can find all the information there. And that's all from our side. I thank you all for your participation today.